Hello students, welcome to EPG Pathshala. I am Dr. Savita Yadav from AIMS. Today we are going to talk on module Affinity Chromatography, Principle, Methodology and Application from paper Techniques in Molecular Biophysics 1. So what are the objectives of this module? What are we going to learn from this module is theory of affinity chromatography, affinity media, column packing, procedure of affinity chromatography, which are the factors which affect affinity chromatography and its application. Now, what is affinity chromatography? Affinity chromatography is also known as bioselective adsorption. This technique uses specific binding affinity between two molecules. In this technique, a specific ligand is immobilized to an inert support matrix in such a way that when sample is passed through it, the molecules which has specific affinity to the ligand will bind to it. After washing non-specific molecules present in the samples, the bind molecules can be eluted from the ligand attached to the matrix. Affinity chromatography utilizes protein's biological structure for its purification. Thus, it offers high capacity, selectivity, and resolution. As a result, purification is often easily achieved with affinity chromatography. So what is the history of affinity chromatography? In 1968, affinity chromatography was introduced by Affinson, Meyer and Pedro, and they have described a method to purify an enzyme using immobilized substrates and inhibitors. This field is still developing and nowadays widely applied to explore fields such as post-translational modification and protein-protein interaction. In recent decades, the development of mass spectrometry instruments and their coupling with the reverse phase affinity chromatography has helped enormously in disease biomarker discovery. The principle of affinity chromatography is solely based on the reversible interaction between the target molecule and affinity ligand immobilized on the column matrix. The suitable ligand selection is determined by the inherent recognition site present on the target molecule in such a way that the binding between target molecule and the ligand is very specific as well as reversible. Affinity chromatography process has three basic steps: sample loading, washing, and non-specific molecules and elution of target molecule. Prior to starting a separation process, affinity column, a column which contains affinity resin, a specific ligand, immobilized on the inert matrix, is equilibrated with the conditions that favor maximum binding between target molecule and affinity ligand. Sample is then loaded onto the column, followed by column washing to remove unbound substances so that only target molecule is attached to the affinity column. Finally, the target molecule is eluted either by changing the buffer condition such as pH or ionic strength that destroy and the interaction between target molecule and affinity ligand by adding a competitive molecule which has more affinity to ligand. Figure on your screen shows the process of affinity chromatography. It shows simple steps like loading, washing and elution. In loading, the sample is passed through the ligand attached to the column matrix. Once ligand attaches the target molecule, the other non-specific molecules are removed by washing. Now the column has only got target molecule and your ligand. Now target molecule can be eluted from the column by using appropriate buffer which destroy the interaction between target molecule and the ligand. There are several components of affinity chromatography which are decisive for purification of specific biomolecules from complex mixture. The important components are column matrix, spacer arms and affinity ligand. Now let us discuss about column matrix. Traditionally used resins for affinity chromatography are agarose, cellulose, polyacrylamide and silica. The commercially available resins come in wider range of particles and their pore sizes. Also, some affinity ligands resins are also available with commonly used affinity ligand immobilized on them. For example, protein A or G, heparin, sibacrin blue, etc. In addition to above mentioned porous matrices, non-porous and monolithic support matrices are also being developed these days. Affinity ligands are now coupled to various supports such as latex beads, nano and macroparticle beads, magnetic particles, microarray surfaces, and many other supports to capture specific biomolecules. The factors which should be considered while selecting an appropriate affinity support material may include pore size, stability, chemical inertness, and mechanical stability. Therefore, an ideal matrix should be mechanically and chemically stable, should be easily derivatized, it should have a surface for easy ligand accessibility, and it should have good flow properties, and it should be stable to long range of buffers. Another important component is spacer arms. A spacer arm, which is shown on the screen, is usually inserted between support matrix and affinity ligand that facilitates proper binding with target molecule. The spacer arm is helpful in providing a more effective binding environment. A spacer arm is critical because in many cases binding sites of a target molecules are buried in the structure and thus restricted for ligand access. Also, it is useful when a receptor area of target molecule is too large for ligand. Thus, addition of a spacer arm helps the affinity ligand to move away from the surface of the matrix, which allows it to bind target molecule more efficiently. The length of spacer arm is critical as improper size may cause binding failure or in some cases a non-specific binding. Figure on your screen shows the schematic representation of how spacer arm is attached to the matrix. You can see there is a matrix, and to matrix appropriate spacer arm is attached, and to the spacer arm ligand is attached, and to that ligand target molecule binds, so that there is enough space for the target molecule to bind. So spacer arm is important because it is providing enough space for the target molecule to bind. Different kind of ligands are used to separate target molecules, including antibodies, diligand, DNA, RNA, and peptides. In addition to commercially available affinity ligand matrix resin, customized affinity supports can also be designed to target unique biomolecules. To design costume affinity system, a method is required to covalently attach a ligand to matrix. The type of linkage between the immobilized ligand and the matrix is critical as it is significantly affect the performance of the affinity separation. Immobilization of affinity ligand to the support matrix is independent of type of application, which is facilitated by well-characterized chemical reactions through common chemical groups. Prior to immobilization of ligands, the matrix is first activated by an easily reactive compound that is capable of binding with ligand functional groups. The common reactive compound chemistries are primary amines, carboxylic acids, and aldehydes. Then activated complex generates a covalent connection between the affinity ligand and support matrix, and this results in immobilization of the ligand. The type of connectivity is very vital as improper linkage may adversely affect the separation process. If it blocks the ligand structure, it may reduce the quality of affinity separation. Weak linkages may allow the release of immobilized ligand from the matrix and may contaminate the purified molecule. Also, if a chemical reaction introduces a charged functional group into the matrix, it may result in non-specific binding, and thus it will generate ion exchange effect. When immobilizing a ligand on solid support matrix, and care should be taken to ensure that immobilized ligand is able to bind to the target molecule easily and actively. Ideally, ligands are immobilized on a matrix in such a way that its binding pocket are fully exposed to bind the target molecule. Activity of ligand is generally affected by some factors including orientation of ligand, multi-site attachment, and steric hindrance. Ligands can also be immobilized by several methods, including covalently coordinated with metal ions, adsorbed on the surface, or by entrapping inside a pore. However, covalent immobilization and adsorption are most commonly used strategies. An affinity ligand specific for a target molecule can be attached covalently to a solid support matrix in case of covalent immobilization. Covalent immobilization is one of the most widely applied methods to attach an affinity ligand onto a matrix. Various coupling chemistry for immobilization of ligand covalently to the matrix may include amine, aldehyde, carboxyl, carbonyl, hydroxyl, and sulfhydryl groups, which can efficiently link ligands onto a support matrix. Another strategy for immobilizing ligand is adsorption. This is used for immobilization of specific ligand on an affinity matrix, which might be specific or non-specific. In specific immobilization adsorption method, attachment of ligand is site-specific, and the common examples are avidin or streptavidin are used for adsorption of ligand containing biotin. Another example is protein ANG, which is used for immobilization of antibodies. While in case of non-specific adsorption, no specific sites are involved and ligand molecules are adsorbed on the matrix surface by hydrogen bonding or hydrophobic forces. In specific adsorption method, the primary ligands such as streptavidin, avidin, protein ANG are first immobilized on the solid matrix using covalent immobilization, that is amine reactive method. Biotin or an antibody as per the need is then attached to them through adsorption. Now let us see what are the types of affinity purification. Based on the different types of affinity target, various affinity purification strategies are applied. Commonly available affinity chromatography procedures are: first is antibody purification. Antibodies are usually purified from serum or culture supernatant by applying affinity chromatography method in two particular ways. The selection strategy is basically dependent on specific requirements, such as for what purpose, an activity assay or detection method, antibody can be purified will be used. Another is affinity purification with immobilized protein AG column. Protein A and protein G are known to bind most of the IgG subclasses. IgG is produced by most of the mammals in response to various immunogens and is known to be the most abundant immunoglobulin. So these proteins are widely applied to capture antibodies using affinity strategies. Immobilized protein A and G resins are available commercially. A specific antibody purified with this method might be contaminated with other IgGs or similar molecules if present in the crude sample. So this method allows only partial purification of antibodies. Another method is affinity purification with immobilized antigens. To purify a specific antibody from a crude sample, this strategy is applied. Antibody-specific antigen is first covalently immobilized on a solid support matrix, and then crude sample is passed through it. A purified antigen is usually a hapten or a peptide that is inserted to the host animal as an immunogen to induce an immune response and thus production of specific antibodies. Captured antibodies is then eluted using appropriate buffers. Various activated resins and ready-to-use affinity kits are available commercially. Pull-down assays are affinity methods commonly applied for analysis of protein interaction, specifically protein-protein complexes. To begin with a pull-down assay, a purified protein is essentially required. This purified protein is known as bait. It is used to pull down its interacting protein in the sample. Interacting protein or proteins are commonly termed as prey. The initial protein can either be purified from natural sources or it can be generated through cloning and expression. The bait protein is first immobilized on solid matrix and it is then mixed with the crude sample containing interacting proteins. The bait-prey protein complexes are finally recognized using various available methods. In antigen purification using antibodies, antigens of interest can be purified at small scale using antibody affinity chromatography. This is commonly referred to as immunoprecipitation. In this method, specific purified antibody is covalently immobilized to the matrix. A typical immunoprecipitation method involves separation of an antibody-antigen complex using protein A or G resin. In first step, an antibody is incubated with the sample, which binds to specific antigen present in the sample. And next, antibody-antigen complex is captured by protein A or G beads. Finally, antibody-antigen complex is eluted using appropriate buffer, preferably by lamellae buffer and separated by gel electrophoresis. Co-immunoprecipitation, it's commonly referred to as co-IP and is very similar to immunoprecipitation method. However, this method is used for studying protein-protein interactions. It involves not only capturing of antibody-antigen complex, but also the proteins attached to the antigen in the crude sample. In contrast to IP, a soft buffer is used in co-IP, which is not so harsh that it destroys protein-protein interaction. A buffer with no extreme pH range and less detergent, usually less than 100 millimolar, is preferred in case of co-immunoprecipitation. Avidin is a protein that has good affinity for biotin. Biotin is a cofactor which is known to have crucial biological role in eukaryotes. Streptavidin is another protein that binds to biotin. These biotin binding proteins have 4 biotin binding sites, which make their interaction ideal for affinity purification. Avidin biotin interaction is the strongest known non-covalent interaction that is unaffected by extreme pH ranges, high salt concentration, temperature, organic salts, and denaturing agents. Also, bond formation between biotin and avidin is very rapid. Other than affinity purification, avidin-biotin affinity system is widely applied in Western blotting, ELISA, immunohistochemistry, fluorescence-activated cell sorting, electromobility shift assays, and cell surface labeling. In the process of recombinant protein expression, a functional domain, which is called as a fusion tag, or some additional amino acids are usually added to help in its purification. The fusion tags are usually appended to DNA, which encodes the specific protein sequence. For example, his tag is one of the most common fusion tags. It can either be a short string of 6 histidine residues or 9, that is polyhis tag. These tags easily bind to metal ions like cobalt, nickel, etc. Other important fusion tags are GST, HA, flag, MBP, SUMO, etc. Unlike histidine and GST tags, epitope tags require specific antibodies for purification and are rarely used for purification at large scale. Lectins are carbohydrate or sugar binding proteins, usually containing 2 or more carbohydrate binding sites. Lactin affinity chromatography uses this sugar binding property to purify glycoproteins. It is a powerful method for studying post-translational modifications in protein, specifically glycosylation. In lectin affinity technique, sugar, which is N-linked or O-linked, is attached to a protein bind to an immobilized lectin. The bound glycoprotein is then eluted using a buffer containing sugar of higher affinity. Several lectins are available commercially. Concavalin A and wheat germ agglutinin WGA are widely applied lectins for purification of glycoproteins. Each type of affinity system have its own set of condition and specific challenges. There are many factors that affect the separation quality in affinity chromatography, but the most important factors are selection of affinity ligand, buffer systems, and sample preparation. Now, how selection of affinity ligand will affect your purification? Suppose you have taken a ligand which has affinity not only towards target molecule, but it has affinity towards some other molecule also. So what you want to purify with this ligand, you will not be able to achieve that kind of purification using non-specific ligand. So ligand should be very specific for the target molecule. Now, orientation of ligand is also important. If ligand is oriented in such a way that it is not able to bind your target molecule properly, so you will lose your target molecule in the process. So, selection of affinity ligand for any kind of affinity chromatography is very important. Another important system is buffer system, the buffer which you are going to use. Buffer should be such that both the molecules, your ligand as well as the target molecule, should be stable in this buffer. And this buffer should be such that it should not affect the interaction between ligand and target molecule. So, interaction between ligand and target molecule should be maintained, and both the ligand as well as your target molecule should be stable. Third important factor is sample preparation. Suppose sample you have prepared, it has precipitated, you have taken highly concentrated sample. If you pass that highly concentrated sample through your specific ligand, it may not be able to bind properly to that ligand. So sample should be diluted, it should not be precipitated. If it is precipitated, again it will create problem for affinity purification. The immobilized affinity ligands to target biomolecules are widely used in several industries and research setups. The common application for affinity-based chromatography technique is in depletion of high abundant proteins. Suppose I take an example of researchers who are doing proteomics of serum. And they want to work on low abundant protein. They want to find out some biomarker. And as we know, in serum, albumin is highly abundant protein which can mask other low abundant proteins. So affinity chromatography is applied to remove albumin from serum samples so that a researcher can focus on low abundant protein present in the serum. Similarly, suppose if you want to enrich proteins from a source. Suppose one person is working again on some fluid, and then from that fluid he want to take out only glycoprotein. He want to enrich glycoprotein samples. He want to use glycoproteins for some other research purpose. So these again, by using lectin affinity chromatography, you can enrich proteins by using affinity chromatography. Another example is study of protein-protein interaction. Some person has say purified certain protein, and we all know that protein-protein interactions play important role in biological function of any protein. So interacting partners of that specific proteins can be separated by using affinity chromatography, by using co-immunoprecipitation. And most common use is purification of antibodies and proteins from the any source. So these are the applications. You can use affinity chromatography for depletion of high abundant protein, enrichment of protein, to study protein-protein interaction, and purification of antibodies and proteins. So what are the precautions one should take while doing affinity chromatography? First is selection of appropriate media, and affinity ligand is very essential. Buffer should be selected in such a way that it does not interfere with the affinity between ligand and biomolecule. It should be capable of maintaining natural environment needed for their interaction. Sample should be prepared in appropriate buffer, and it should not be very concentrated, as it may hinder the affinity due to overcrowding of other molecules. So students, let us summarize what we have learned from this module. Affinity chromatography is a technique that uses specific binding interaction between molecules. Affinity chromatography has three main components. First is solid matrix, second is spacer arm, and third one is affinity ligand. Immobilization of ligand on solid support can be achieved by various methods such as covalent immobilization and adsorption. There are various types of affinity separation system. These are antibody purification, IP and co-IP, Lactin affinity, Biotin affinity, and recombinant tag purification.